I have the pleasure of introducing Dr. Wilmore Webley, Associate Professor and Associate Dean, Office of Inclusion and Engagement in the Graduate School at the University of Massachusetts Amherst. Dr. Webley has received a Master's of Science and Ph.D. in Microbiology at the University of Massachusetts Amherst with expertise in immunology, infectious disease, host-pathogen interactions, and a concentration in vaccine development. He will present on the new bivalent BA.5 booster and the challenges the new variants impose. Dr. Webley, you have the floor. Thank you.
So hopefully you're all seeing my screen. I was asked to talk about the new COVID variants and what really is a bivalent vaccine and what that means. I'll do a little bit of preamble to get us there. Just to remind everybody that COVID is still a pandemic and is still affecting all of us. We're just over 6 million deaths. It's a significant number of deaths from any pandemic and still ongoing in the US alone. Last month, we had just under 10,000 deaths in the United States. So just in case people think, okay, this is something that's gone and we no longer have to deal with it. But we've known about coronaviruses for a long time, since the 1960s, as a matter of fact. And initially, they were just studied as cold viruses. And then 3 times we've had coronaviruses come into the human population and have been deadly. And the first one, as you probably remember, was the SARS outbreak in 2000, 2002, 2003, infecting about 8,000 people, killing just under 800. And at that point, of course, they started in earnest to study these viruses with knowing that there was this opportunity for outbreak. They're called coronaviruses because they have this really neat characteristic of having this crown-like spike proteins on their outer surface. And the reason talking about the spike proteins is that they're central to the vaccines that have been developed. And in 2002, when the initial SARS outbreak happened, it was then that they actually started to look at making vaccines against coronaviruses. Of course, because SARS went away so quickly after they had slaughtered the civet cats and broken the transmission cycle, they weren't able to test any vaccines that were in development. And then, as research typically goes, the money kind of dried up. People lost interest because we just didn't see SARS, what we know now as SARS-1 or SARS anymore. But then in 2012, MERS popped up, the Middle East Respiratory Syndrome coronavirus. And that now brought coronaviruses back to the forefront to show us that this was a family of viruses that was just not going away. And then, of course, research again restarted. Lots of people, including the NIH, started looking at vaccines for MERS and for other coronaviruses. People like Dr. Peter Hotez started again looking at vaccines. And so what I'm showing you here is a scanning electron micrograph of an infection with SARS-CoV-2. And it's infected a cell here. And what you're seeing is the blebbing, is literally what you're seeing in my background, of this cell. This cell is dying by programmed cell death. And then all of these little yellowish dots that you're seeing here, these are the coronavirus. Turns out that it takes about 10 hours for you to have a productive coronavirus infection. And it produces tons of progeny, which renders the cell pretty much crippled in the presence of this type of viral infection. Now, as I said, the virus is known by the spike protein that you see on the surface. And we talk about it as just one protein, but it's actually a trimer. It's actually 3 proteins that come together to form what is now the receptor that binds to our cells in order to get the virus in. In other words, these proteins on the surface of the virus are the keys that the virus uses to unlock our cells to get inside of it. And what it uses on the surface of our cells is a protein called ACE2. This is angiotensin converting enzyme receptor 2. And these are receptors that are ubiquitous on the surface of our cells. They're on the respiratory cells, but we now know that these receptors are all over the body, from liver cells to lung cells to pancreatic cells, kidney cells. All of them actually have ACE2. And so once these cells have ACE2 and some other core receptors, the virus is able to enter. As you saw there, released into the cytoplasm of the cell, the RNA, positive strand RNA, and that leads to the production of new viral proteins. That's why viruses infect our cells. They need to make more of themselves. And in order to do that, it has to use the cell's machinery or ribosomes to be able to create the proteins that it needs. And it will do this for as much as it can, for as long as it can. Its goal is to make as much progeny as possible. It's not the goal of the virus to kill us. It's the goal of the virus to be as transmissible as it possibly can, so it can make as much of itself as it possibly can. And so in the process of doing this, making more and more of itself, viruses make mistakes because they're copying the entire genome. I'm going to play a little movie for you that kind of depicts it.
Every time a virus infects a person, it uses their cells to make copies of itself, replicating this complex code again and again and again. But eventually, it makes a mistake. Sometimes it deletes or adds a letter, sometimes it flips them around. That mistake is called a mutation, and it slightly changes the instructions for making the virus. That slightly altered virus is a variant. Since viruses are constantly going through this copying process, it's normal for them to change over time.
And so the interesting thing about large viruses like coronaviruses that we're talking about, SARS-CoV-2 specifically, is that they have a proofreading system in them. They have a spell check, so that if a mistake is made, they correct that mistake. And so it doesn't make half of the mistakes that influenza makes, for example, but it makes enough so that it can be problematic overall. And so the process of mutation is happening at pretty much a specific rate, because we know it has a proofreader. We know it has a protein in there that checks these bases to make sure that they're in the right order. But every now and then it makes a mistake. So even when your spell check is on, when you're writing a document, the larger the document, the more words you write, the more mistakes you make. This is a very large virus by all means. And so the longer that a virus sticks around in a person, the greater the probability that it's going to have these changes, these mutations. And so for a person who is immunosuppressed, for a person who has underlying conditions that make it hard for them to fight any sort of infection, that person is going to have more viruses in their system to deal with, and they're going to have the virus in their system for a longer period of time, which means the probability of these variants popping up is going to be higher. And that's important. In other words, if you are unvaccinated, and you're also immunosuppressed for whatever underlying condition, the probability that if you get infected, a new variant will develop in you is greater, right? It doesn't mean that you're going to start a new wave of a new deadly variant, because there's a lot that goes into that. It has to escape our immune system and all of that. But sufficing to say, SARS-CoV-2 has been able to do that repeatedly. So this pandemic started with what we call the Wuhan strain, the original strain of the virus. Again, here's a cartoon depicting your spike proteins. I think each virus has probably 70 or 80 spike proteins. That spike protein is what the virus uses to bind to the angiotensin converting enzyme 2 receptor. So it can enter the cell, also binds to co-receptors. And if you have mutations, clearly the mutations can't happen at that binding site to the extent that they will prevent it from binding. If that happens, then that variant is going to die out. However, what can happen is that you could actually have mutations that make that binding tighter. It could make the binding looser, which means that the variant is not as infectious as its parent strain. But if it's binding tighter to the receptor, to the ACE2 receptor, then that variant is going to have abilities that its parent strain didn't have, right? And so you've seen here that we've had alpha, beta, gamma, delta, and then we came to Omicron. And then Omicron shows us exactly what we can have with variants. We can have subvariants. So we had BA.1, BA.2, BA.3, 4, and then for the past several months, we've been dealing with a BA.4, 5 variation. But what the general public hasn't been privy to is the fact that for each of these subvariants, there are sub-subvariants and sub-sub-subvariants of those. It's just that many of them haven't risen to the point where they become variants of concern. Because here's the issue, that any infecting virus can have random mutations. And those mutations could possibly make the virus more fit. But if it never gets transmitted to another person, then nobody ever really knows about it, right? If it doesn't get transmitted, it's not a problem. And so transmission becomes important. And transmission is going to be dependent on how well that spike protein, that trimer protein, is still able to bind to the ACE2 receptor here. That's really important in the overall scheme of things. And so what we have, what the WHO has developed, is this scheme for looking at variants and categorizing them. And so you have your variants of concern, which is what your BA.5, BA.4 variants are, your Omicron, Delta, Alpha variants. They're variants of concern, and there are some characteristics of variants of concern. A variant of concern is a variant that has the potential to have increased transmissibility, and that typically means that it's able to bind probably more tightly, more readily to its receptor. It could have increased virulence, and it's either because something else in the virus has changed besides just the receptor. It could have other mutations that make it more virulent, cause more severe disease, or that could lead to a decrease in the effectiveness of therapeutics like the vaccines. What we saw is that our original vaccine, monovalent vaccine against the Wuhan strain, wasn't as effective against the Omicron variant. That alone makes it a variant of concern. What we've seen is that the monoclonal antibodies that used to be used at the beginning of the pandemic, when Omicron came around, none of them were very effective, right? That becomes an issue because it cuts down on the number of options you have for therapeutics. So the variants of interest also can't be ignored because these are variants with genetic changes, and we now know the combinations of amino acid changes that are relevant to make a variant that we have to keep an eye on. And so we've seen those variants as well. They have significant community transmissibility. They just haven't emerged yet as a global public health issue. And then, of course, you have variants that are constantly being monitored because, again, they have these mutations or combinations of mutations that make us keep an eye on them. They just haven't posed any risk yet. Doesn't mean they're not going to pose future risks. So the next thing I'm going to show you will probably blow you away a little bit, because here I'm showing you the family tree of the variants that we've had so far. And as you can see, we had the Wuhan strain. This is the original Wuhan strain, from which came the Delta variant, from which came the Alpha variant, from which came the Beta, the Mu, the Lambda, the Gamma variant, and from which came the Omicron variant. So what I'm showing you is that all of the variants that we've had so far have come from the original parent strain. Why is that significant? Because here's what happened with Omicron. Once we had Omicron, then our BA.2, our BA.4 and 5 variants actually came from this subvariant. That could happen with every single one of these, right? In other words, people are looking, say, okay, this virus has just varianted out. It's exhausted the amount of variants it can have. It's not even anywhere close. Because just on this limb, this branch of the Omicron, you're seeing all the subvariants we have. But wait, look at this. This is the current map that scientists track, right? This is just the Omicron tracking. BA.1, BA.2. From BA.2, we got BA.4 and 5. From BA.4 and 5, what they're tracking now are those variants of concern and variants of interest and other variants that we're keeping an eye on, because right now, your BQ11, which you probably haven't heard about, and your BQ1, as well as your BE virus, your BR2, your XBB, right? XBB1, XBB4, these are the variants that are tracking in other countries as well as in the US. But you haven't heard anybody talk about it. So all of these that you're seeing on here, all of these are sub and sub-sub variants of Omicron variant. So this now shows you what I'm talking about, that you're having mutations in these viruses, literally almost every replication. And every time that happened, there is the probability that you could create a new variant of concern, a variant that is escaping the vaccines that we have. And that is why you have to update vaccines. So let me show you now what is circulating and what we're seeing in the US. So this is the US right up until this week. And what are you seeing? That in September, you're having mostly BA.4 and 5, right? That was what was dominating. Now, I want you to look at November, because here we go. You're still having some BA.5, but look at this. This is BQ1, BQ1.1. BQ1.1 is a derivative of BQ1. Then you have this BF7, right? And so what you're seeing over here is that, yes, BA.5 is still dominant, but it only accounts for just under 30%, just around 1/3 of what is being transmitted. Look at what is bringing up the next end. It's the BQ variant. In fact, if you actually add the two BQs, that actually gives you 44%. They're nearly accounting for 50% of what we have right now. And these are variants that are more immunoevasive than BA.5, right? From your original Omicron variant. And then you have all of these lineages down here that you see are popping up in some percentage amount, your BF and your BA.2 and your BA.2.7, which for a while we thought would take over. I guess the reason why I'm showing you this is that I'm just saying to you that what you see typically reported in the popular press and what is actually happening, what scientists are sequencing and seeing every day, are really two different stories. You catch the ones that are really out there, but underneath you're seeing. So what is happening globally? I wrote this statement out. For the first time during the pandemic, there are more than 300 subvariants that are circulating, and none of them is dominating globally. So I show you this case of South Korea, where they're having probably about 20 or 30 different variants that are vying for control. You see these highlighted in bold. These are the major ones. And again, you're seeing the BQ1, the BQ11, one of the ones I didn't show up there is the XBB1, which is another variant of concern that everybody's looking at. And how we look at them is we look at how often they're doubling. The ones that are doubling at a faster rate are the ones that we take into great consideration. But what you're seeing currently is that you're seeing a hotspot in Western Pacific and in Southeast Asia. And again, in South Korea, what you're seeing is something that we haven't seen here. Even with the increase in the B1 variants, we have not seen an increase in hospitalizations in the United States, which is good news for us, but we're seeing it in South Korea. So when we look, this is the CDC wastewater testing over the last several months. And as you can see, in this last couple of weeks, last 6 months, you're seeing the data in the last couple of weeks, what we're seeing is that we're seeing an increase in the West, we're seeing a decrease in the Northeast, and we're seeing an increase, a little bit of uptick in the Midwest United States in terms of total number of cases that we're seeing in those regions. So are we going to get a terrible winter with COVID? There's nothing here that says to me, definitely yes, but there is that possibility. And again, as I said, all it takes is a new variant. Now, we talk a lot about immune response to this virus. Remember that the vaccines that we have, both the Pfizer, the Moderna vaccine, pretty much most of the vaccines on the market are made from the spike protein, right? It's mRNA that codes for the entire spike protein. And that is what we get vaccinated with. Why? Because the spike protein is what the virus uses to get into the cell. It's an important protein to the virus. And if the virus doesn't have that protein, then the virus can't get into the cell, right? So that's why it was targeted. It was known for years now, before SARS-CoV-2 came about, that that's how you would try to block a coronavirus. All of the vaccine iterations were looking at the spike protein. Now, we see most of the times when we think about an immune response, we think, okay, the body sees the virus and it makes an antibody against it, or it has a T-cell response against it. And that's not exactly true. And if you're going to understand why we need to have bivalent vaccines, it's important to get this concept. What I'm showing you here is a rather large hotel, right, in this image. And when the body sees the spike protein of the coronavirus or the coronavirus itself, what it sees is literally analogous to a large hotel. And when it looks at it, it is not seeing the entire hotel as a single entity. What it is seeing is that each of these tiles that you're seeing here on the outer side, these large tiles, you have antibodies that are built just against that one tile. You have antibodies that are built against these ledges. You have antibodies that are built against these windows and these little balconies. You have antibodies that are built against these main doors. And these specific B cells that are responding, for them, if they're responding to the door, they don't care about this window. For them, the window doesn't exist. They're just making a response against the door. So in essence, what you have is what we call a polyclonal response, where you have hundreds or thousands of antibodies being built against a large protein like a spike protein. So when we say that these variants are evading the immune response, it doesn't necessarily mean that no antibody in the system is binding to them. It probably just means that these important antibodies that probably block the doorway and neutralize the virus now, the new variant has changed this door. So instead of it just looking like this, it now runs the entirety. Instead of you having this ledge looking like this, these windows now become round. And so the antibodies that were made against the square window no longer recognize it because the windows have changed and these ledges have changed. Maybe what the mutation has blocked out this area. So instead of having this indentation and having these open windows or closed windows or curtains, they look different. And so the response of the immune system is different. In other words, the memory cells that were there that remembered this architecture, once you change that architecture and change the color and change the shapes, the vast majority of those antibodies have no function anymore. And that is what reduces the response that you're having. I hope you appreciate that. That's really important to understand. So, for example, what I'm showing you here is a little diagram of what we call antigens. Antigens are the binding sites on an entity that antibodies bind to. And I'm showing you them in different shapes and different colors. And this antibody that is induced by antigens is only binding to this yellowish orange antigen because that's the only shape that it recognizes. It could care less about this blue, purple, red, orange, green one because it doesn't recognize that. That is essentially what is happening with the immune response that we made against the original Wuhan strain when it comes to the Omicron strain, okay? So here's a depiction of a bacillus. Here are the different proteins on its surface, what we call antigenic determinants, and the antibody A that's made against this antigenic determinant doesn't recognize antigen B. It takes antibody B to recognize that. Right? And if you had a mutation where antigen B shapes becomes differently, it's now an octagon, then antibody B would no longer recognize it. So the antibody would be there, but it would be of no use because it's not being effective. So essentially what you want in terms of a vaccine for SARS-CoV-2 is that you really want an antibody that binds to the spike protein, neutralizes it. By neutralizing, I mean blocks it, so that it never binds to the ACE2 receptor, right? Once you have antibodies binding to all of these 70 or so spikes around the surface of SARS-CoV-2, then it can never enter a cell because it's been blocked, physically blocked from doing that. And so again, as I said, the receptor binding area of the spike protein, you see, all of these are depicting different types of antibodies that bind to the spike. And the virus has the ability now to change just one amino acid. And then this antibody doesn't recognize it anymore. Doesn't recognize this site. And it changed another amino acid over here, and this antibody doesn't recognize. But all of these other antibodies are recognizing the protein itself. It's just not neutralizing it as effectively because everything can change. In fact, for this receptor binding domain, you can't really change in here too much because if the virus does that, then it'll never bind to the ACE2 receptor. So here's another analogy. Here is the SARS-CoV-2. These are the spike proteins on its surface, right? So there are your spike proteins. Here you have the human cell with the ACE2 receptor. This is what the spike protein binds to. So think Legos, right? And then here's the antibody that's made against it. This is the neutralizing antibody. And look at what the antibody does. It binds to the spike protein so that now the spike protein gets blocked and it can't bind. However, if you develop a mutation, the mutation now takes you from this square to a rectangular shaped spike protein, for example, and now it fits better, literally locks in onto the ACE2. But now this antibody has no effect, can't neutralize anymore because you can't bind to this different shaped protein. And that's essentially what I'm talking about. Now, we have examples of current bivalent, multivalent vaccines. So their current flu vaccine is a multivalent vaccine. In fact, it's a quadrivalent vaccine. And by that, I mean, it's being made against 4 different strains of the flu virus. You have 2 A strains and 2 B strains. Of course, if you remember this from your microbiology classes, it is your A, influenza A subtype, that is the main ones that cause disease in humans and cause pandemics and outbreaks. But of course, your influenza type Bs can cause serious diseases, especially in young kids. So they put all of those together, whether the egg-based or the tissue culture-based vaccine, quadrivalent vaccine, right? And with these, they're putting whole viruses in. They're growing them up in eggs and they're putting them in. But Gardasil is a 9-valent vaccine, right? Because you have so many different variants of human papillomavirus. So we know, for example, that 6 and 11 are the main ones that cause wart. 16 and 18 are the main ones that cause cervical cancer. The others cause other types of throat cancer and different warts. So they've just added those. Initially started out as a quadrivalent vaccine. Now it's a 9-valent vaccine. So vaccines with multiple valences have been around. They're known to us. And so what we're looking at now with the bivalent vaccine for COVID-19, here's the setup for the mouse study that was done for that. Here's the original Wuhan strain. And when they injected the original Wuhan strain spike protein into mice, you get a really strong response against the original Wuhan virus, but not very good response against, in this case, let's say Omicron virus. You see the reduction in the amount of antibodies that are binding? That reduction signifies that it's not recognizing different sites on the virus. Same thing, that if you take now this mutated Omicron virus and you injected the spike protein of it into a mouse, the mouse will have a robust response against the Omicron strain in this case, but not against your original Wuhan strain, which means that if I just vaccinate you with the original Wuhan strain, you're vulnerable to Omicron. And if I just vaccinate you against the Omicron variant, you're susceptible to the original Wuhan strain and probably to alpha and beta. Okay, so in order to make you as protected as possible, you put in a bivalent vaccine, 2 spike proteins from the 2 different variants, your original Wuhan strain and your Omicron. And now you have protection against these 2 strains. And the data shows that. So this is the clinical trial data. And hopefully you can see this graph. But what you're seeing here is your original vaccine, right? And this is in people who had no prior infection. And this is before, and this is 29 days later. And this is antibody level. So essentially what you're having is that you're seeing that when you vaccinate, you're getting an increase in individuals who were previously unvaccinated. And same thing with the new bivalent vaccine, only to a greater extent. And then, of course, if you look at individuals who had prior infection, you still see that increase, but the bivalent vaccine, again, give you a much higher increase. And when we look at symptoms, in other words, adverse effects from the vaccine, there's no difference between the bivalent vaccine and your original one wild-type vaccine. And so here's a little video explaining the boosters for this strain.
The updated vaccines have 4 primary advantages compared to the original vaccine. One is that they retain protection for the original strain. The second is that they provide updated protection, better protection against the currently circulating strains, BA.4 and BA.5. The third is they do induce a broader immune response that is expected to provide better protection against future variants. And the fourth is it does provide a boost to make sure that everybody does have up-to-date immunity going into the winter season. So these updated COVID boosters contain mRNA that encode for spike protein for two different strains of the virus. One is the original strain that was in the original vaccines, and the other is updated, so it includes the spike protein for BA.4, BA.5. And for each vaccine, both Moderna and for Pfizer, each vaccine contains half of each, half of the old formulation and half of the newer updated formulation. You know, studies have shown that by using this bivalent approach, you get a broader immune response, and this might even help protect against future variants. So not only will we retain protection against the original strain and get updated, better protection against currently circulating strains, but this may even protect against future variants that we haven't seen yet. Anybody who's 12 years of age and older who's at least received the primary series of vaccination is eligible to be boosted. So this includes people who've just had the primary series, people who've had the primary series and one booster, or people who've had the primary series and two boosters. It has to be at least 2 months since the last vaccine dose. And we do this to make sure that we optimize the immune response. For those who have had recent infection with COVID, it's best to wait 3 months after the infection to get this new booster formulation. Even though COVID is currently circulating at less high levels compared to previous in this year, we still have 500 people a day dying in the U.S. from COVID. So it still is a serious threat to people's health. This boosting program is expected to save between 100,000 and 150,000 lives over the next 6 months. So the adverse effects following immunization with the new booster are very similar to what we've seen with previous doses of the vaccine. There's no new adverse effects or side effects that are going to be expected. We still continue to expect some people to have a sore arm for a day or two following immunization. Some people may have a fever or other systemic side effects, such as tiredness. These generally go away in 1 to 2 days.
So I'm going to end here by just saying that, again, we have to be cognizant of our environment. We have to look and see, where am I? Am I in a crowded area? What is the rate of cases that we're having in my area right now? And take the necessary precautions. Of course, do get vaccinated and boosted with the bivalent vaccine so that you can have maximum overall protection, especially as we go into the winter months when we're encountering other things like RSV and influenza. And I'll stop there.